All right, we've been talking about how you make proteins, but how do you stop making proteins and then start up again if you need them? We don't want to make all proteins all the time. We want to be able to make them when we need them and stop making them when we don't. It's really important for cells to control the expression of their genes, to be able to control when proteins are made and when they're not. Now, it was really much easier to figure this out looking at prokaryotic or bacterial cells because their systems are so much simpler than eukaryotic cells. So we're going to talk about what scientists first figured out looking at prokaryotic gene control, and then we will talk about it with eukaryotes, which is a bit more complex. We won't go into that much detail though, so we won't overwhelm you. But let's talk a little bit about how to control your genes and what they're producing, the proteins they're making in bacteria. Bacteria, like anything else, need to respond quickly to their environment. If they've got enough of something that they've been making, they need to be able to stop making it because it would be wasteful to continue making it. But if they don't have enough of a product, or if they find a food source and they need an enzyme to digest it, they need to be able to start up making their enzymes or whatever they need to make. They need to be able to stop production and start production of different proteins when they need them and when they don't need them. It's just common sense. Now, there are ways to do this that don't necessarily involve genes. We talked about this with cell respiration when we talked about phosphofructokinase as an allosteric enzyme that could shut down the pathway if we had enough ATP being made. Well, there's all kinds of enzymes like that. For example, this isn't the cell respiration pathway, it's a different one, but it looks very much like glycolysis. But here our end product is an amino acid called tryptophan. And if your cells are making tryptophan, but your cells determine you've got enough of it, tryptophan can feed back into an allosteric enzyme here, cause the enzyme to change its shape so it no longer receives the substrate here, and then the whole pathway shuts down, okay? And so we stop making tryptophan if we don't need it. But the reality is that the genes that are making all these enzymes are still going to be making them. We just won't be making tryptophan anymore. Wouldn't it be more efficient if we could also stop making all these unnecessary enzymes as well, and then start up that process again when we do need the tryptophan, maybe later, okay? So, if we regulate at the gene level over here at the DNA instead of just at the enzyme level, we can even be less wasteful because we won't make a bunch of enzymes that we don't need, okay? Now, generally speaking, cells can control the amount of enzymes, proteins they're creating by regulating whether or not the genes that code for those proteins are going to get transcribed or not. We can turn the genes at the DNA level on and transcribe them, or we can turn them off and not transcribe them, not make RNA, okay? So if we turn genes off, if a bacteria has enough of that amino acid tryptophan and it doesn't need to make the tryptophan, but it also doesn't need to make the enzymes it uses to build tryptophan. So we're going to turn all those genes off. But we can also, if we need the tryptophan, we can be able to turn those genes back on. Or, for example, if a bacterium all of a sudden finds itself in an environment with a sugar it wasn't using before, like lactose, it can turn on the genes that make the enzymes to digest lactose because it's got a new environmental signal. So there's all kinds of stop and go signals that a cell could use to shut its genes off and turn its genes back on again to be very, very efficient in its use of resources. So bacteria tend to group their genes together into little units with related functions. And this is called an operon, okay? It's a group of genes that have a related function. Perhaps all of these genes might be being used to make tryptophan, for example. Okay? So all the enzymes in a particular pathway might be clustered together in the DNA. Now, also part of the operon is the promoter. That region, the Tata box, if you'll remember, that tells RNA polymerase that is in charge of transcription where it needs to get started, okay? One promoter controls the transcription of all these genes. So here's this promoter region on this DNA for bacterium, and here's the genes for example, making the enzymes that help make tryptophan, okay? And this promoter would be where the RNA polymerase would bind, and it would just transcribe all of those genes, okay, if we needed them. All right, an operator is also part of an operon, and the operator is actually a place where we can shut things off. It is a place where something called a repressor protein can bind to the DNA. All right, so let's kind of get a little bit more info on that repressor protein, okay? A repressor protein would would bind to DNA at this operator site. Now notice that the operator, which is in yellow, comes right after the promoter, which is where RNA polymerase would bind. So it basically just blocks the way. It blocks the way. It's like an elephant sitting on the DNA, and it's not going to let anything through. And that means if that operator is occupied with a repressor protein, then we're not going to make any RNA as long as that's sitting there. Okay? So let's look at this. Let's say we have some genes here. Perhaps they're involved in making tryptophan. They're enzymes. This gene is for an enzyme. This one's for an enzyme. This one's for an enzyme. This one's for an enzyme. And all those enzymes are part of something, a biochemical pathway to make tryptophan. Okay? The RNA polymerase would bind at the promoter Tata box on the DNA. Okay? And it would start moving down the gene and transcribing it, and we would get an mRNA molecule. And in that mRNA would be the instructions for making these 4 different enzymes, okay? But if that repressor is in place, then we're not going to be able to send the RNA polymerase down to do its job, okay? It will block that RNA polymerase from making mRNA, and so we won't make the enzymes. Let's look at an actual example of this, and we're going to go back to that tryptophan example, okay? So now these are the genes for enzymes that would be in the pathway to make tryptophan. And what we're going to do is RNA polymerase would bind at the promoter and move on down, and we would make the mRNA that would make that would code for all those enzymes that would help to make tryptophan. Okay? So there's all the tryptophan we end up making out in our cell as a result of these enzymes being active and functional. Okay? But suppose we have enough tryptophan. In that case, if we have excess tryptophan, there might be enough tryptophan present to bind to the repressor protein. And that would cause the repressor protein to take a shape that allows it to bind to the operator. Okay? So when it binds to the operator, then that RNA polymerase may come in, but it's not going to be allowed to bind and do its job. It's getting blocked. Okay? And that makes sense because clearly we have enough tryptophan. We don't need to keep running this pathway. We don't need to keep making these enzymes. So that's a helpful bit of gene control. Okay? So this is just showing that tryptophan itself, the product of the pathway, is an allosteric regulator. It goes into a second active site in this repressor protein, causes it to change its shape so that it can bind to the operator, and then we're not going to make any tryptophan that we don't need. Okay? That's called a repressible operon. It's getting repressed. It's going to always be on, but it can be stopped. Okay? Now, let's look at one that is called an inducible one. One that is generally off, but we can cause it to turn on. Generally speaking, bacteria might not be around lactose sugar a lot, but if it does find some, it would like to eat it. Okay? So, normally, the enzymes to create, to digest lactose will be inactive. The genes to make those enzymes will be inactive. They will be repressed because normally lactose sugar is not around, and so why make the enzymes to digest it if there's no lactose sugar around? But if there is lactose sugar around, okay, there is lactose sugar, it will bind to the repressor in this case and cause it to change its shape to lift off of the operator, and then RNA polymerase can come in and do its job and make the enzymes to digest all that yummy lactose, and then the bacteria is happy and can digest that sugar. Okay? This is called the lac operon. It was one of the very first that was ever figured out how this gene control was achieved, and it controls the production of 3 enzymes involved in digesting lactose. Okay? So lactose, again, is a regulator of that repressor protein, and if it binds to the repressor, it will cause it to lift off the operator region so that the RNA polymerase can work. And these are the guys who kind of figured out about this operon stuff. So in summary, repressible operons usually function when the cells are using pathways to make something, and then these repressible operons will shut it down. Inducible operons are usually in catabolic pathways where we find things that we want to break down in the environment, digesting things, and usually they're off unless we find the thing in the environment we want to digest, and then they get turned on. The lac operon is a great example of that. All right, that's gene regulation in bacteria. We will take a very quick look in eukaryotes, and then we will be done. Take it easy. See you next time.